Hey, and welcome to the last big chunk of lectures for the semester. So in the last lecture, we talked about animal diversification, and we talked about the various structures that different classes of animals have, and then how we categorize or organize those animals based on particular characteristics. And then from this lecture going forward, we'll be discussing animal physiology. So in this particular lecture, we'll start with some basics of physiology, and then we'll move on to talking about homeostasis and the excretory system. And this is chapter 21 in your textbook. Physiology is the study of complex functions carried out by organs and organ systems in living organisms. And this definition basically sums up the rest of the semester. So we'll be taking a look at various organs and the tissues that make up those organs, and those organs, how they function and how they work together in organ systems to allow for an organism to function properly. And something to always keep in mind as we're going through the next couple weeks is form follows function. And that basically means that in order to perform a specific function, the cells, the tissues, the organs have to have a particular form. And so your textbook gives a example of this on a macro level of all the creatures that swim through aquatic media have a streamlined body plan. And that's because the function is to swim through aquatic media, so they all have similar forms, which is the streamlined body plan that allow them to do that. We can also see that in tissues, and we'll see that in this lecture in a couple of slides, how different tissues are structured in a particular way to perform a specific function. And a lot of times we see similar forms in various animals because of a similar selective pressure that has because they have to perform a similar function, right? So similar selective pressure requires a shared function, which means that they will have a common form. So just remember, form follows function, and keep that concept in mind as we're going through the rest of this semester. And animals exhibit levels of complexity, and all these levels are hierarchical to one another. And so the base level of complexity are cells, and then cells make up tissues, tissues make up organs, and organs make up organ systems. The simplest layer in animal biology and like the first kind of level of the hierarchy are the cells. And cells, as you may remember from 1103, are the smallest unit of life. They're also the smallest unit of organization. So cells are not haphazard, they're not random, they're highly organized, highly specialized units that work individually or as a collective to form a particular function. And some examples of cells, there are many out there, but just a few that you may be familiar with are neurons, gametes, red blood cells, stem cells, bone cells. The list goes on and on and on. There are various different types of cells that make up the animal body. The next level of complexity are tissues, and tissues are a group of similar cells that work together to perform a specific function. And so as we're going through the next couple slides, we're going to be taking a look at various tissue types, and I just want you to keep that concept of form follows function in your mind. And so as we're looking at these various tissues, take a look at the cells that make up those tissues and how all those cells are performing the same function, so they have the same or very similar structure to one another, as well as take a look at the tissues and how they're structured. Each tissue type performs a specific function, and so they are structurally laid out so they can best perform that function. So just keep that in mind as we're looking at the various tissue types. And there are four types of tissues that we've seen in animals. We have connective tissue, epithelial tissue, muscular tissue, and nervous tissue. Connective tissue is composed of cells embedded in a large extracellular matrix. And the extracellular matrix, or matrix, if you're not familiar with what this is a kind of like a meshwork of non-living extracellular material. And this material is derived from the cells that reside within that matrix. So those cells within the matrix are constantly pumping out different material, mostly polysaccharides and proteins, but some other nutrients and polymers and things like that as well. And they're pumping them out into their environment around them. And then once those components get out into the environment, they polymerize into various different structures that make up the matrix. And so a good example you can see here in this top picture are these kind of circular, oblong shaped, more rounded shaped structures are the cells, and these are called fibroblast cells. And they are pumping out polysaccharides and proteins that make up this kind of meshwork that's around them. So all of this fibrous looking material here, like all these kind of like stringy looking stuff, is the matrix. And you can also see it down in this lower picture here as well. All of these blue circles are the cells, and everything that's not that blue circle that's bright colored, so all the red and the green, are actually extracellular matrix as well for those cells. And this matrix can vary in structure. So for some parts of your body, the matrix is more liquid, like in the case of blood. Sometimes it's more solid, like in the case of bone. And sometimes it's what we call semi-solid. And semi-solid is kind of like jello or jelly. It's not quite a liquid, not quite a solid. So if you can just imagine like jello or jelly type material, that's kind of what that extracellular matrix is like for that type of connective tissue. And the matrix provides support and structure and protection for the cells that reside within it. But as a whole, connective tissue provides similar benefits for organs and for the entire body, provides shape, structure, and support for the body. And it is connective. It holds everything together. That's how it gets its name. So I mentioned that in the previous picture that was up at the top, that those were called fibroblast cells. And fibroblast cells are like the star connective tissue cell. These cells are really good at pumping out making two types of fibers. So they secrete collagen and elastin, which are major components of connective tissue. And so this cartoon here I really like because they show you kind of the extent of how much collagen and elastin that these fibroblasts can make. So in this picture, all these purple dots are representing the um, fibroblasts, and then all of the larger material here, like this one, is collagen fibers, and all of the purple kind of more mesh work, the thinner pieces, are the elastin. And so these fibroblast cells, there aren't as many of them as you would expect, but there's such a wide variety and amount of elastin and collagen to make up that matrix. So they're kind of like the stars at making connective tissue. And they're present in almost all connective tissue, with the exception of blood. So they're not present in blood because blood has a liquid matrix, and collagen and elastin are solid fibers. Connective tissue also provides a location for anchoring, and it helps to facilitate the communication between cells. So the connective tissue helps to also hold cells in close proximity to one another, and that allows for them to communicate well. It also allows for them to transmit signals from one another to another. And they also play a major role in growth and healing. So if you ever cut yourself, that first layer of tissue that forms after you've started to heal, after the blood clotting has been removed, that first layer is the connective tissue, and it plays an important role in providing that scaffold so your epithelial tissue can grow on top of that. And some examples of connective tissue are tendons, ligaments, adipose tissue, bone, blood, you name it, it's probably a connective tissue if it's not a nervous tissue, muscular tissue, or epithelial tissue. So as I was just mentioning, any tissue that's not muscular, nervous, or epithelial tissue is connective tissue. So with that being said, you can imagine how many different tissue types fall underneath this category of connective tissue. And so to break up this diverse group of tissues, we have two major categories. And then underneath those categories, we have some subgroups. So the first major category is connective tissue proper. And connective tissue proper is referred to as the packing material of the body. It's the tissues that we normally would think of as connective tissue. And so we'll talk about some examples in a minute and it'll make a little bit more sense. So underneath connective tissue proper, we have loose and dense forms. Loose connective tissue proper is called that because its matrix is semi-fluid, semi-solid, and it's very flexible. And it's like this because the collagen fibers that make up the loose connective tissue are relatively spaced apart, which allows for more fluid in between those collagen fibers than if they were closely packed together. And so if we were just to draw an illustration as an example, let's say we had a section of connective tissue. The collagen fibers in a loose connective tissue would be kind of like this, right? They're highly spaced apart to allow for plenty of liquid in between the collagen fibers. And the role of loose connective tissue is to provide cushion, lubrication, and insulation for the body. And so some examples are the connective tissue around your organs that holds everything together and holds your organs in place, as well as the connective tissue that's underneath your skin. So the loose connective tissue under your skin is the reason why you can do this to your skin and it not just like fly off when you pull your skin, right? The connective tissue is holding your epidermis to the rest of your body so you don't end up falling apart. And then also adipose tissue, which is fat tissue. And this is essential for your survival. And many of us will figure out how essential adipose tissue is once it gets winter time because it provides insulation for the body, but it also provides cushioning for your body as well. And then you also have your dense connective tissue. And dense connective tissue is called dense because the collagen fibers in it are far more tightly packed. So these tissues are much stronger. So if we were to draw an example of this, instead of the collagen fibers being loosely packed like we saw above, they would be very, very closely structured to one another, right? So there's far less space in between these collagen fibers, which allows it to be more dense and highly packed and strong. And some examples of this are tendons and ligaments. And the responsibility of the dense connective tissue is to hold bone to bone and muscle to bone. So it needs to be there to provide that support. So if we didn't have that, if I were to extend my arm without the connective tissue holding everything together here, without these tendons, I would extend and then my arm would just fall. It helps to keep everything together, right? It also attaches my muscles to my bone so they're not sliding past each other and things like that. So it provides that support I need to have a proper structure. We also have special connective tissues, and these are connective tissue that you may or may not immediately think of as connective tissue. And they're special because they usually have some sort of special cell type that makes up the matrix of these types of tissue. Special connective tissue can be rigid or solid, rigid or fluid. An example of rigid is bones. So your bones provide protection and support for your body. You can imagine the protection as your rib cage, protecting your lungs and your heart from damage, as well as the bone that makes up your spinal cord is also providing structure and support. And then also in structure, we have a skeleton. Without our skeleton, we would be a large mass of just like goop and skin. It would be like a mess. So the connective tissue of our bone helps to give us the structure that we have. And then the matrix of bone is highly dense because it's undergone a process called calcification. And you don't need to know that term. Just know that this is essentially when an organism will incorporate calcium into its matrix to give it more structure and support. So this is why you need calcium in your diet so that you can have the calcium to add into the matrix of your bones so that they're strong and not brittle. Then we also have cartilage, and cartilage is not nearly as rigid as bone, but it does still also provide flexibility, support and structure. So we talked about cartilaginous fish And we talked about here in your ear, you can feel those hard bits that are also flexible, and that's cartilage. And so this is clearly strong because it can be a whole organism skeleton, and we can feel it too in parts of our body, but it's also flexible. And then also in vertebrates, it also provides a cushion where your joints are located. So this becomes a problem when those cartilage cushions break down, and that's how you can get arthritis. So, like in your wrist here, there is cartilage that is between the two bones that keep them from rubbing against each other. But once that cartilage breaks down, you now have bone rubbing against bone, which is very painful and causes inflammation that gives you lots of pain during arthritis. And cartilage is composed of collagen, elastin, and protein, all bound to these long carbohydrate fibers. And so that's how you get that strong structure and support, because we have the collagen and elastin and proteins like we see in regular connective tissue, but they're also onto these tightly bound carbohydrate chains to provide even more structure. And then finally, we have our blood. And blood is an example of a liquid special connective tissue. It's composed of red blood cells, white blood cells, and platelets. RBCs is red blood cells, and WBC means white blood cells. And the matrix of blood is actually the plasma. So plasma is primarily composed of water, proteins, and carbohydrates. And this makes up the matrix that surrounds your blood cells. Next, we have epithelial tissue. And epithelial tissue is a thin layer of tissue that covers every surface inside and outside of your body. And you can see this as the most apparent example as skin, right? We can see skin covers our body. But then there's also epithelial tissue that makes up the protective layer of your blood vessels and of your gut and your intestines. All of your organs have epithelial tissue that surrounds them and protects them. The epithelial tissue is composed of epithelial cells that are very, very tightly packed together. And they're so tightly packed together that they're essentially air and water tight. So if any liquid or if any gases want to pass from the outside environment or pass between a particular environment to get inside of your organs, it has to pass through this barrier of closely packed cells. And these cells are closely packed to one another because they have various structures that basically anchor them to each other. So some examples are like tight junctions, which you can see in this cartoon here, which directly connect the one cell to another. And you can have things called desmosomes, which are almost like protein bridges that connect the cytoplasm and plasma membrane of one cell directly to the cytoplasm and plasma membrane of another cell. So there's a lot of different types of anchoring mechanisms that help to keep these cells in close contact with one another. And all epithelial tissue have kind of two sides. So there's different types of epithelial tissue out there and different tissue cells. We're not going to talk about each of those in depth or anything like that, but just know that some epithelial tissue is one layer of tightly packed cells, and then some epithelial tissue can be multiple layers of tightly packed cells. In either case, there are always two sides. There's the outer side that faces the environment or the cavity. And so an example of this are the outer layer of your skin, right? It's facing the environment. There's also the part of your gut lining that faces the part of your stomach lining that faces the acid is also the outer layer. Then you have the inner layer, and the inner layer is responsible for connecting that tissue to the underlining layer of tissue. So you can see that in this picture here, it's showing you that this would be the outer part here that's in facing the environment, and this would be the inner side, and it's anchoring to the extracellular matrix. Similar thing if you have multiple layers. One layer will be facing more towards the outside, and then one layer will be facing more towards the inside, and it will be responsible for attaching that epithelial to the underlying tissue or tissues around it to keep everything in place. So epithelial tissue can play a couple of different roles. It can play roles in protection, obviously. So we talked about how skin protects our bodies, as well as the epithelial tissue that makes up the gut helps to protect the rest of your body from your stomach acid. So this one is an example of that. It's showing you how there's a lining of cells that make up your stomach lining that keeps your acid from leaving your stomach and then eating up the rest of your organs. So the layer of tissue cells is actually this here, the layer of epithelial cells, and then there's a layer that connects. This layer here would be responsible for connecting to the basal layer and keeping everything in place. Epithelial cells can also play roles in transport. So the epithelial tissues that line your blood vessels are responsible for allowing for carbon dioxide to enter into the veins, to be carried away, to be removed, and for the transport of oxygen out of the blood vessels into the tissues that need the oxygen. And then also can play a role in secretion of different things from glands. So like your salivary gland, epithelial tissues are responsible for making the saliva that allows you to eat your food and things like that. I included this picture as well here because it's a good example of showing you how the form follows function here. So the epithelial tissues are all the tissues that are within this kind of circle here. And you can see how they all are epithelial tissues and they all have a similar structure. And their structure is different from the cells that reside within the center of this tissue. And same thing with the epithelial tissues, epithelial cells that line on this outer side of this tissue sample as well. They have similar structure to the epithelial tissues that are here because they have a similar function, so they have a similar form. And some examples, before that, the epithelial tissues are commonly exposed to adverse conditions. So like your skin is exposed to heat and dryness and getting scraped and rubbed off, as well as your gut lining is exposed to the acids. Your intestinal lining is exposed to the movement of food and getting scraped off and things like that. So they're in some conditions that are generally not favorable, so they often get damaged, so they're frequently replaced. The tissues that make up your mouth as well are very commonly replaced. And then some examples of some epithelial tissue, some we've already talked about, your skin, your gut lining, intestinal lining, blood vessel lining, etc. So then we also have your muscular tissue. And muscular tissue is composed of these long contractile cells that have these protein filaments in them. And the protein filaments slide past one another. And when they slide past one another, they generate a force that causes, that results in movement. So we're not going to go through how muscles contract in a whole bunch of detail, but I did want to cover some basics so everybody understands the general gist of what's happening when muscles contract. So when we have a muscle cell, like I mentioned, it has protein filaments, and there are two types of filaments. You can see them here. You can see this red filament, and then we have this blue filament. Blue. And both of those filaments you can see are attached to a disc. And so when a cell is relaxed, a muscle cell is relaxed, those two discs are farther apart from each other. But when that muscle cell gets stimulated, then that red filament will slide past the blue filament like this to move those two discs closer to one another. So you can see that when you compare the relaxed to the contracted, which is down here, the lower half, the contracted discs on the side are closer to one another. You can also see that if you look at the blue filaments. So the blue filament distance was much wider when the muscle fiber was relaxed than when the muscle fiber is contract, the distance is much shorter. And that's because when they're contracted, now everything's moved closer together and that contraction allows for movement. So that's a general gist of how that works. If you're more interested in learning more specifics about that process and how muscles contract, I recommend looking up some videos on YouTube. There are a lot of good examples and videos on how this process works. You can also take a molecular biology class if you're interested in these types of concepts in general, or anatomy and physiology to learn more about this process. But anyway, so there are three types of muscle tissue. The first we're going to talk about is skeletal muscle. And skeletal muscle is our voluntary muscles. So it's what we think about whenever we say, yeah, I'm going to the gym, getting ripped, gains. All that is skeletal muscle. And it's called striated because it has these like darker bands within it that kind of give it more of a striped look. So you can see that in this cartoon here. They did kind of these bands of darker color, which are like here, right? That's the striation. But you can also see striation down here if you look at this actual image of true muscle tissue, and you can see that striation as well in this muscles. Okay, and these types of cell, I mean, not cell, this types of muscle, skeletal muscle, is voluntary muscle. So we can control this muscle just through our sheer will. And it can be controlled consciously or on an unconscious level. Some examples of an unconscious level are our eye movements, right? You're not really thinking about how your eyes are moving when you're watching YouTube videos or watching this video now, but your eyes are moving subconsciously. Same thing as like your lungs that allow you to breathe. There's also skeletal muscle, but you're unconsciously moving it. You can consciously move it. You can stop breathing, hold your breath, but on an unconscious level, it keeps you alive and keeps you going. And then skeletal muscle is stimulated through neurons. And so we'll talk about neurons a little bit later in a couple of slides. But neurons basically are directly attached to muscle fibers, and they send electrical and chemical signals directly to the muscles. And those signals is what tells the muscles to contract, and starts that process of sliding those filaments past each other to get the muscle to contract so you can get movement. And skeletal muscle is generally, for the most part, attached to bone. So pop quiz, quick question. What connective tissue is responsible for attaching skeletal muscle to bone? Hopefully you can figure that out. And then each of the individual cells within the muscle fiber, skeletal muscle fiber, are called muscle fibers. And so these are multinucleated long cells. So each of these that are shown up here, each of these long tube looking things is a muscle. And they are multinucleated. So they're cells, they're basically really, really long cells that have multiple nuclei. So in that picture up there, these like blue discs looking things are supposed to represent the nuclei. And then the red is the long cell. You can also see that in this picture down here. For this is one really long cell, one really long multinucleated cell. And if you were to look at it, you can count the nuclei, which are the darker colors. So if I had to count, I would say there's 1, 2, 3, 4, 5, 6, 7. It gets a little weird in this area, but maybe 7 or maybe 8 nuclei in just this image of this part of the cell. So there's just multiple nuclei in one really, really long cell that is responsible for contracting to get you to move. Now, this multinucleation is not what we see in cardiac cells, which is what we'll look at next. Cardiac muscle is the muscle that makes up your heart. And just like we saw with skeletal muscle, cardiac muscle is also striated. So we can see that in this picture up here. In their cartoon, they show you the striation, so that banding pattern in the muscle. But you can also see it down here in this photograph of cardiac tissue. If you look really closely, it's a little bit harder to see, but you can see that striation as well. So there's it's easiest to see in this area where you can see this kind of striated banding pattern in this cardiac muscle. And despite that similarity, there are a lot of differences between cardiac muscle and skeletal muscle. For one, we cannot consciously control our cardiac muscle contractions. So you can't just close your eyes and be like, beat slower, beat slower. Like it just doesn't work that way, or beat faster. You can manipulate your heart rate by increasing your level of stress or anxiety or physical activity, or decreasing it by like you doing yoga or sleeping or something like that to make yourself more relaxed. You can influence your heart rate, but we can't consciously increase or decrease our heart rate. Additional differences are the fact that the cardiac muscles, unlike the skeletal muscles, have a single nucleus. And these cells are also elongated, but they have more of a branched mesh-like orientation to them. So if I was to make some alterations to this cartoon above, I would draw maybe defined lines between cells. So let's say that this would be one cell, and then this would be another cell, and maybe this would be another cell, and this would be another cell, right? So these are all elongated branching cells, but they have a single nucleus. And each of these cells has lots and lots of mitochondria. And these mitochondria are really important because your cardiac muscles must be able to contract for your entire life, and they must be able to contract quickly when necessary. So having a lot of mitochondria allows for a lot of ATP to be produced, and ATP is used when muscles are contracting. So if there weren't enough mitochondria in your cardiac tissues, then you probably wouldn't be able to get your heartbeat to continue for your entire life, or you wouldn't be able to do strenuous activities, right? Because your heart rate wouldn't be able to increase because there was not enough ATP to allow for it to accelerate in in the number of beats. So those are some major differences. And if you look at all of the muscle fibers and their orientation in general, you can also see that there's some differences. So when we looked at the skeletal muscles, they were closely packed fibers right next to each other running parallel. But in the cardiac muscles, they have more of a mesh-like look to them. They're not super closely parallel packed muscle fibers. And where these cells are, though, closely packed together, cell to cell. And where these two cells meet is at a location called the intercalated disc. And you can see that in this picture down here. This really dark bands that you see, like this one here, and then like all of these dark bands, are the intercalated discs. These are not the normal striations like we talked about in the skeletal muscle. And these intercalated discs are basically gap junctions between two adjacent cardiac cells, and they allow for the electrical signals to pass between those cardiac muscles very, very quickly. And so what happens when your cardiac muscle contracts, there's a signal sent from a neuron that then sends an electrical signal to a certain set of cardiac fibers. And then those cardiac fibers need to send that signal to all the other fibers that make up the heart so they can all contract at once. And this is how they do it. Without these gap junctions between your cardiac muscle fibers, the signal would move much slower. And so you might get like levels of cells contracting. So you get maybe this level contracts, and then these cells contract, and then these cells contract, which can be problematic. So we all know if you have an irregular heartbeat, that's not good for your survival. That can be very detrimental to the survival of an animal. So these intercalated discs, or these gap junctions, allow for the swift passage of electrical signals through all of the muscle fibers that make up the heart, so they can all contract at one time. The last type of muscle tissue we have is smooth muscle. And smooth muscle is called smooth because it lacks the striations that we saw in the cardiac and skeletal muscle. So you can see that as an example here. If you look at this picture, you don't see that banding striated pattern in this tissue. Instead, this tissue is composed of tightly packed, elongated cells that look something like this. Also, the cartoon up here is showing you kind of these tightly packed, elongated, smooth cells that make up the smooth muscle. And smooth muscle, it is present in the walls of your organs as well as the walls of your blood vessels. And we cannot control the contraction of smooth muscle. So if you've ever had a stomach ache, you're pretty familiar with this. We have no control over when those muscles decide to contract, how fast they contract, and how strong those contractions are. They just happen to us. And so when they do contract, though, there's a lot more rhythmic and slow contraction than what we see in cardiac and skeletal muscle. And so what happens is a good example of this to think about is in the intestines. So as food is being moved through the intestines, your intestinal smooth muscle is contracting, and it'll contract here, then it'll contract here, and then it'll contract here, and it just keeps going down, right? And those contractions is what helps to push the food through your digestive tract, in addition to the epithelial tissues that make up the gut lining. And so just think of that as a good example. To me, whenever I think of smooth muscle, I always think about the peristaltic contractions of the intestines as it moves food through the gut. And the smooth muscles can be stimulated through neurons, like we saw in the striated muscles of the skeletal muscle and cardiac muscle, but they can also be stimulated to contract via ions. So differences in ion concentration can stimulate the contraction of these muscles. So they're not always under neuronal control. Last but not least, we have nervous tissue. And nervous tissue is specialized for the storage and transmission of information. And this information is stored and transmitted in the form of chemical and electrical signals. Nervous tissue also allows us to respond and sense stimuli. So I mentioned before that neurons are directly connected to muscle fibers, skeletal muscle, and they allow for movement, right? So this picture here is an example of that, where you can see all of these long yellow dense fibers at the bottom. These are all muscle fibers, skeletal muscle fibers. And then this branched material here that's coming from the side is the neuron or the nervous tissue. And it's in direct contact with that skeletal muscle. So it can send chemical signals from the skeletal muscle to, from the neuron to the skeletal muscle and cause that muscle to contract. And that's how you get movement. So the neurons sense the stimuli, and then they send that chemical signal to your muscle fibers to allow you to respond to that stimuli. So there are two types of cells that make up nervous tissue. We have our neurons, and the neurons are the excitatory cells. They're the ones that are sending and receiving these chemical and electrical signals. So they're the ones that we normally think of as doing the heavy lifting of the nervous tissue. And they have a couple of different parts. They have the dendrite, cell body, and the axon. The dendrite are these branches that are responsible for receiving information from other neurons. And so they can be seen here. This is an example. This is also an example of a dendrite. These are all dendrites. So all this branching material are dendrites. So if there was another neuron here, and let's say these are the axons of that neuron, and they were sending a chemical signal out, that signal will be picked up by the dendrites of this neuron. And then that chemical signal or electrical signal would then pass from the dendrites to the cell body. And the cell body is where all the other parts of a cell that you normally associate with a cell are located. So the nucleus, the mitochondria, the endoplasmic reticulum, all the protein production all happens in the cell body. From the cell body, the signal is sent to the axon. And the axon is the part of the neuron that's responsible for transmitting the information to the next neuron, or transmitting the information to the muscle fiber. So the axon, in the case of connections to skeletal muscle, would be the part that's directly attached to the muscle fibers. So the axon then transmits the signals from the ends of it to whatever it's attached to. In addition to neurons, there are other cells that play a major role in the functioning of the nervous system. And this is the glial cells. And glial cells can be seen in this image here as these orange cells that are all along the axon of this neuron in this picture. And they're basically like the support staff for the neuron. They provide nutrition, insulation, protection. They regulate the environment for the neuron. Basically anything that the neuron needs, these glial cells do it. So neurons would not be able to perform their function as well as they do without these glial cells. And they're not just present on the axon, they're present all over. So you can see that better in this image here. There are several different glial cells. So the actual neuron is this here, as well as all these dendrites that are extending. These are all the dendrites. And then if you wanted to see the glial cells, all of these guys, oh, let's choose another color. Yeah, all of these dots here are actually the nuclei of glial cells. And the you can't really see the projections from those because it's kind of all over the place. But these are the nuclei. So there are glial cells all over that are supporting this neuron and its neighboring neurons. And the glial cells, they are the support staff, but they do not transmit electrical signals themselves. So they might look very similar to a neuron, but they do not function in the same way. The nervous tissue composes two different systems. So we have two nervous systems. We have our central nervous system, which is composed of the brain and the spinal cord. And then we have our peripheral nervous system, which is responsible for sensing our environment. This is our sensory neurons and our motor neurons, so allowing us to move, basically sensing and responding to stimuli, peripheral nervous system. And they can also control gland secretions. So if you're sensing danger and you get really stimulated, neurons can tell your epithelial gland cells to secrete more mucus or secrete more sweat if you're feeling anxiety. They can play a role in that as well. And some examples are the brain and spinal cord. All right, so getting back to our layers of complexity, the next layer of complexity after tissues are organs. And organs are structures made of tissues that serve specialized functions. And there are multiple tissue types that make up an organ. So organs are not composed of one single type of tissue. An organ will have epithelial tissue, connective tissue, muscular tissue, and nervous tissue all together to function, or they might have different combinations of those four types of tissues. Some examples of organs, we all kind of know some examples of organs, but livers, kidneys, hearts, brains are just a few examples. Finally, we have our highest level of complexity, the organ systems. And organ systems are groups of organs that work together to accomplish physiological functions. And each organ system is a group of organs that work together for a particular function, but all of the organ systems also work together to allow for an organism to live and function properly. So organ systems do not operate in isolation. They all work together to accomplish the shared goal of the organism being alive. And there are 11 different organ systems that are recognized in animals. You can see a list here, some examples: digestive, excretory, endocrine, skeletal, nervous. We'll talk about all of these for the rest of the semester. And this one today, we're going to focus on the excretory system. And then as we go through the semester, we'll talk about how each of these systems functions and how they relate to each other. So now that we've talked about some basics of animal physiology, we're going to actually get into the actual material. So we're going to begin with homeostasis and the excretory system. Every type of animal body has a specific defined range of conditions where it functions best. And if you get too far outside of that range, that animal is in trouble and is in danger of sickness or death. And so, with that being said, animals must maintain a relatively consistent internal environment in order to survive. And this maintenance of ideal environmental conditions is the term homeostasis. And so there are a variety of internal conditions that can change based on your environment, based on resource availability, based on your habits. So some examples are temperature, your blood sugar levels, blood pH, CO2, O2 levels, the amount of salt in your blood. All those different types of variables are apt to change depending on environmental conditions or what you eat that day or what you do that day. And so your body has to use a variety of physical and chemical methods to regulate all these different variables and make sure that you remain within that optimum range. So there are two terms or types of conditions that you'll see for the next couple of slides. And these two terms are regulators and conformers. So for various different variables, an organism can be a regulator or a conformer. It just depends on the animal and the conditions. And so what are these terms? So, like I mentioned, the environmental conditions can change. And a regulator, no matter what the change in the environmental condition, they are actively maintaining that optimum ideal internal condition. So they work to maintain homeostasis. A good example that I thought your book had was in this picture here. So this fish is a regulator of how much salt is in its body. So whether it's in a low salt concentration environment, like let's say a freshwater environment, or if it's in a high salt concentration environment, like a marine environment, regardless, it works to maintain a constant amount of salt in its environment. So if it's in a freshwater environment, it makes sure it maintains more salts. And if it's in a salty environment, it makes sure it gets rid of excess salt because it wants to maintain this optimum salt concentration in its body. And then you have conformers, and conformers allow for their internal environment to fluctuate with environmental conditions. And so they used an example here of conformers with temperature. So this same fish is a regulator for salt concentration in its body, but it's a conformer for temperature. So if it gets in the winter and the temperature is low, then its body temperature will also be low. But in the summer, let's say the water temperature is higher, it will allow for its body temperature to also be higher. So this is a good example of an organism being a regulator for some variables and a conformer for others. And you'll see this type of regulator, conformer type of way of maintaining homeostasis in the next few types of slides. And so there are a couple of ways that an organism can help to actively maintain homeostasis or passively maintain homeostasis. And one of them is through extracellular fluid. So I mentioned before that your cells are pumping out different metabolites and to make a matrix. There's also a fluid that is surrounding all of your cells, and this is called the interstitial fluid. And it fills the space between cells and surrounds the cells and keeps them in this like nice aquatic environment. And it's composed of obviously water, nutrients, and then it also has waste products. So as your cells are getting rid of waste, it goes to this interstitial fluid and then is eventually moved to the circulation to be removed by your bladder and liver and kidneys and things like that. And so the interstitial fluid actually is pretty substantial. It's about 1/3 of all the water in the human body is this interstitial fluid. So it might seem like a minor thing, but making sure you have this liquid is really important to keeping your cells hydrated and alive. And it can act as a nice buffer for conditions as well. So if conditions change very slightly, they're out of the optimum kind of set point, the interstitial fluid can help balance out that changes so you don't fluctuate when there's even minor changes in the environment. Another method that animals can use to maintain homeostasis is through the use of feedback systems or feedback loops. And there are two types of feedback systems in animals. We have negative feedback loops and positive feedback loops. Negative feedback loops are the most prominent type of feedback system, and this is basically when a certain environmental stimulus or change in the environment will stimulate a system of events that will counteract that change in the environment to move everything back to the optimum range. And so there are two major components in this system. You have your sensors, which sense the change in the environment, and then you have your effectors. And the effectors are the elements that are working to counteract that change in the environment. And so your book gave a really good visual that I think is helpful to understanding this concept. It doesn't really relate so much to the body, but it's a good way to understand how negative feedback loops work. So they use the example of the thermostat and AC and furnace in your home. So the thermostat in your home is the sensor. So it senses the temperature in the room. So let's say I tell my thermostat that the optimum temperature for me is between 72 and 75 degrees. So we have 72 to 75. This is obviously degrees Fahrenheit. This was degrees Celsius. You'd be a melted puddle. So let's say in the room, it's in the winter and it gets cold and the room gets down to 68 degrees. Then that sensor, the thermostat, will sense that change and it'll say, ooh, this temperature is below the optimum, and it'll send a signal to the effector, which is the furnace, to turn on to heat up the room. And the furnace will keep running until the room is back to the optimum of 72 to 75 degrees. And then the same thing on the opposite end. So let's say it's a hot Georgia summer day and the room gets to be 80 degrees. The thermostat will sense that change in temperature and send a signal to the effector, which in this case would be the air conditioner, to turn on to pump cold air to the room so we can return back to the optimum of between 72 and 75 degrees. So this is how this system works. It's very similar to how the system works in your body. If your body gets too hot, your body will sense that it's too warm, and your brain will send signals to your body for your sweat glands to start to sweat so you can cool yourself off. Same thing if you get too cold. Your nerves will sense that, hey, it's too cold, and your brain will send signals to your body to shiver so you can warm up. Similar things with blood sugar level. If there's too much sugar in your blood, it will stimulate your cells to take in that blood from your bloodstream using insulin. If there's not enough blood sugar, then signals will be sent to your pancreas to release glucose from the pancreas to keep you going. So these are those negative feedback loops, acting to counteract a change in the environment to return back to that optimum range. On the opposite end, we have the positive feedback loops. And when you have a positive feedback loop, the effector is working to exacerbate the change in the environment. So instead of going back to the optimum range, we're moving further and further away from the optimum. And these positive feedback loops are highly regulated. So they're usually a small part of a larger negative feedback loop. If they're not regulated, you can see how they can get out of hand and cause serious problems for an animal if you're constantly getting further and further and further away from the optimum conditions. And so a good example of this is that your book gave is for blood clotting. So if you get an injury, let's say you get cut doing something you ain't supposed to be doing, and your blood vessels get injured. So this will cause the recruitment of platelets to that area. And platelets are responsible for helping with the clotting. So platelets will travel to that area of injury and they'll stick to that blood vessel to keep it from hemorrhaging. Once it's stuck, those platelets will release chemical factors. Those chemical factors then travel through the bloodstream and recruit more platelets to come to that site. And then once those more platelets bind, they release more chemical factors to require more platelets. So this process keeps going and going and going, where you're recruiting more and more and more platelets until the blood vessel has been repaired. So until the hemorrhaging has stopped. Once that has stopped, you don't want to keep recruiting platelets because that can cause a blockage in the blood vessel. So there is a negative feedback loop that says, okay, no more platelets being recruited, and counteracts this positive feedback loop to prevent more platelets from being recruited, to prevent the platelets that are already there from putting out chemical signals to recruit more platelets. And then it will also start to actively break down that blood clot so that your rest of your body can start healing the injury. So this is just a good example of a positive feedback loop that is within a larger negative feedback loop that is definitely beneficial for you, but you can imagine how if a positive feedback loop was out of control, how that could lead to serious problems for an animal. So I mentioned before, there are a lot of variables that animals have to maintain within a particular range in order to survive and function properly. And one of the major ones is your body temperature. So the regulation of an animal's body temperature is referred to as thermoregulation. And it's important to have your body temperature within a particular range for, number one, just to keep you alive. If your body temperature gets too hot or too cold, that can kill you. But it's also important because the enzymes and cells and everything in your body function best at a very specific temperature range. And so if it gets even just a little too hot or too cold outside of that range, then your enzymes, which do the vast majority of the work in your body, will not be able to function properly. And then that means the cells will suffer, and the tissues and organs and the whole organism will suffer because of that. So you might not die, but you can get very, very sick. Also, having an optimum temperature range allows you to resist being frozen to death, or allows you to be able to have some mobility in the time of day that you do your daily activities. This is particularly important in endotherms because we are able to maintain our body temperature. We are able to go out in the winter and do things without freezing to death. So there are two types of thermoregulation. I just mentioned one, endotherm, and then the other is ectotherm. And we talked about these two concepts in the last lecture. We talked about endotherms are able to harness and maintain the heat that we get from cellular respiration to keep us warm. But ectotherms have to maintain their body temperature based on how much time they spend in the sun and the ambient temperature around them. So to hammer that concept in, we're just going to take a look at this graph here. So it shows you the differences between a cat and a lizard. So the cat is an endotherm, and it's showing you that the cat's body temperature is maintained around 40 degrees Celsius no matter what happens to the environmental temperature. So if it's cold outside, like if it's 15 degrees Celsius outside, the cat's internal body temperature is around 40 degrees Celsius. If it's 35 degrees outside, then the cat's body temperature is still maintained around 40 degrees Celsius. But for the ectotherm, like this lizard here, that's not the case for them. So if it's colder outside, the lizard's internal body temperature is colder. And then if it's warmer outside, their internal body temperature is warmer. So they're highly impacted by the temperature outside. So one of these would be a thermoregulator, and the other one would be more of a thermoconformer. So figure out if you can figure out which one would be the conformer and which one would be the regulator. So being an ectotherm and being an endotherm also has some drawbacks, right? So being an endotherm is great because you're always maintaining a constant body temperature, so your body is able to function at optimum at all times. But the drawback is if the ambient temperature gets too far out of a particular range, we have to expend energy to return to that normal state, to maintain that normal state. So that shows you here in this upper graph that when the cat is in an environment that is between about 32 and 37 degrees, so when they're in this thermoneutral zone, then the cat doesn't have to expend not that much energy to maintain their body temperature. But if it gets too cold, so if it gets colder than around 32, or if it gets too hot, like hotter than 37, the animal has to expend ATP, expend energy to get back to that neutral zone to maintain their body temperature. But the ectotherms, they don't have to do that. They don't have to expend nearly as much energy to maintain their body temperature because their body temperature fluctuates with the environment. So if it gets a little too cold, the environment's too cold and they can't maintain it, they spend a little bit of energy. And same thing if it gets too hot, but you can see the gap in the amount of energy that the cat has to spend at like 39 degrees Fahrenheit to maintain optimum to remain within that optimum temperature range compared to the lizard, which is an ectotherm. In addition to endotherm, ectotherm, there are two other terms that are important to know, and these are homeotherms versus heterotherms. So homeotherms maintain a relatively constant temperature all year round, all the time. And then heterotherms have a fluctuating body temperature. So homeotherms are normally what you would think about like us, right? No matter if it's in the winter or the summer or spring or morning or night, our body temperature is the same all the time. But the fluctuating body temperature of ectotherms is an example of heterotherms, but also hibernating mammals. So when a mammal hibernates, they reduce their body temperature all the way down to usually around ambient temperatures. And hibernation obviously happens in the winter, so their body temperature decreases drastically at some point throughout the year. So a hibernating mammal would be an heterothermic endotherm. But a non-hibernating mammal like you and me or cats and dogs would be a homeothermic endotherm. And then something like a lizard or a turtle would be a heterothermic ectotherm. So keep those concepts in mind. Play around with them a little bit. It might be a little bit hard to understand. Just remember endotherm versus ectotherm, which we talked about. And then homeotherm is maintaining a relatively consistent body temperature all year round, day and night, winter, spring, summer, fall, whereas heterotherms, their body temperature fluctuates to ambient temperatures at some point throughout the year. And there are drastic impacts on your body's function if your body temperature gets outside of that optimum range. So if your body temperature gets too high, we call that hyperthermia. And hyperthermia can result in organ failure and death. So it's very, very serious. We've all heard of heat stroke. If you're outside and you get too hot, your body is unable to cool itself down properly due to the fact that it's too hot outside, or it can be like if you're in the South here, it's too humid outside. If it's too hot or too humid, your body can't properly evaporate the sweat. So you just maintain that heat and you start to basically cook on the inside. And that's what causes people to pass out in the summer when they're doing too much strenuous activity or spending too much time in the sun. And if this goes on too long, you can end up dying. And then on the opposite end, we have hypothermia, which is when you have an abnormally low body temperature. And this can happen if you're outside in very, very, very cold winters for too long, as well as if you jump into like an ice over lake or something like that. That temperature, that cold water and cold air just leaches all the heat from your body and cools your body temperature down way below its optimum range. And this can result in a loss of coordination, difficulties moving, disorientation, an irregular heartbeat, and even death. So you want to make sure that you're not getting too hot or too cold. There are four main ways that heat is transferred between objects. And these objects can be two biotic objects, like in the case of two animals, or it can be between a biotic and abiotic factors, like an animal and something that's non-living in its environment, since we're talking about animals. And so the first one is conduction. And conduction is the transfer of heat through direct contact. A good example of this is if you were to put a heating blanket on your body. So that heating blanket is hot, and then once you put it on your body, you feel that warmth. And that's because the direct contact between the blanket and your skin allows for the heat to be transferred from the blanket to you. Another good example is if you were to give a friend a hug, and that friend is really warm. You feel that warmth from your friend's skin because of that direct contact between your skin and their skin. So that heat from them is being transferred to you. Next, we have radiation, and radiation is heat transfer between objects without direct contact. So a good example of this is if you go outside and especially in the summer and you feel the sun's rays on your skin. This is an example of radiation, right? You don't have to touch the sun to feel the sun's warmth. You can just feel it because that heat is being radiated from the sun and then being absorbed or received by you. Another example is if you sit next to a fire, right? You can feel the warmth from the fire without touching it. That's radiation. Then we have convection, and convection is heat transfer to a medium as it passes by another object. So that medium is aquatic in some form, so it's either liquid water or in the form of air, because air is made of water. And as the air passes by the object, it takes the heat with it. So the heat is transferred from that object to the air. A good example of this is why your skin becomes flush. So your skin becomes flush because your body is getting really, really warm and it wants to release some of that heat. So it increases the amount of blood that circulates to your skin. And then as the air passes by your skin, it takes that heat away from the blood and away from your skin so that you feel cooler. Another example is if you stand outside and the wind is blowing. The reason why wind feels so cold is because as the air is blowing past you, it's taking the heat from your body and taking it elsewhere. So now you feel cooler. Then the last form is evaporation, which is what most of you guys are most familiar with. And this is the loss of heat as a liquid gets turned into a gas. And so an example of this is when we sweat, right? So we sweat because we want to cool ourselves off. And we do this by creating a kind of a liquid barrier water on our skin, right? And then as that water from the sweat is converted into gas, it absorbs basically the heat from us to turn into a gas, and then once it's a gas, it takes that heat away. So that's why we feel cooler, because of the evaporation of the sweat from our skin. And there are a couple of other factors that animals have that impact their ability to regulate their temperature. There are some physical factors such as the animal's body size, their surface area, and the amount of insulation they have. So a good example of insulation having an effect, if you look at a seal pups have a lot of blubber, which gives them a lot of insulation, so they can swim through Arctic waters. In comparison, we as humans do not have blubber. So if we get in those same waters, we'll get hypothermia and die, right? So this helps the seal pup regulate their body temperature in super Arctic waters, but we don't have that. So we wouldn't be able to regulate our body temperature nearly as well in those types of conditions. We also have behavioral factors. So we can change our behavior to allow us to better regulate our temperature. So some examples, if you're outside and you get entirely too hot, what is the first thing you do? You go to the shade or you come inside to the AC, right? That's a behavioral modification to help you regulate your body temperature. Ectotherms do the same thing. If a lizard gets too hot, it will move itself from the sun and move to the shade, or if it's too cold, it'll go to the sun to sunbathe, so it can increase its body temperature. Then we also have some physiological and cellular components that have an effect on an animal's ability to regulate its body temperature. And so this is basically differences in how your body undergoes certain processes. And so some examples, like your book mentioned, are membrane leakiness and endotherms. So it's thought that endotherms have leakier membranes that allow for more ions to come into the cell, which the cells have to then pump out because you don't want too many ions in your cells. And as the cells are pumping out those ions, they have to expend ATP, which releases heat. And that's how we're able to keep our bodies so warm. Another example is increasing the number of mitochondria. So as you're making more ATP and as you're breaking down ATP, it releases heat. And that's another way to keep yourself warm. Other examples are when your skin is really, really hot. Obviously, you can undergo flushing like we just talked about. You can also shiver when you're really cold. This is a way of your body trying to keep you warm, because your brain sends signals to your body that says you need to shake in order to increase the amount of muscle movement that you have, which will increase the amount of body heat that you generate. Then we also see something similar in the brown fat oxidation in babies. So their brown fat is not like the white fat that we see in adults. Brown fat is actually broken down directly by the baby's cells, and so that provides them with the heat that they need to survive in adverse conditions. Another major process that animals have to undergo in order to maintain homeostasis is osmoregulation. And this is the regulation of water and salt contents in your body. And so some of the solutes that we regulate are sodium, chloride, potassium, magnesium, and calcium ions. We also regulate sugars and various other macromolecules to make sure that we have just the right solute to water balance in our cells and in our tissues. And so this is just a clear water in versus water out type of situation. So some ways that we can get water in is either, being actively drinking water. You can get water through eating. You can get it also from absorption and cellular respiration. And then water out is through like urination, defecation, evaporation from sweating, or osmosis. When you're talking, your mouth gets dry like mine is right now. That's another way to kind of losing water that way. And then you, in order to undergo osmoregulation, you don't actually regulate the water, you regulate the solutes, because water will follow the solutes. So if you're still confused about how diffusion works, I really recommend that you go over that material again as a refresher to remember diffusion, active transport, passive transport, all those things, because that's going to be really important to understanding the excretory system. So we have our osmoconformers and osmoregulators. Just like we talked about before, the osmoconformers allow for the amount of solutes in their blood and in their tissues to be to match those of the environmental conditions, whereas the osmoregulators make sure that their solute concentration is regulated so they don't have too many salts or too few amount of salts in their tissues and in their body. So a good picture of this, going back a couple of slides, is when we first start talking about homeostasis and the fish image there. And so this is a big issue for vertebrates and invertebrates that live in salt versus fresh water, right? So invertebrates that live in salt water, they generally will just be osmoconformers. They allow for their body solute concentration to match that of the ambient water. But if you have vertebrates that live in aquatic environments, and saltwater environments, they're more osmoregulators. So they make sure that they don't get too much salt in their body. Same thing in fresh water as well. The vertebrates in fresh water make sure that they don't lose too many salts so that they can actually survive properly. So there are a couple of different structures that help us with osmoregulation, and these are all referred to as excretory structures. And we're going to talk about two in this lecture: the Malpighian tubules in insects and the kidneys in vertebrates. And an improper balance in osmoregulation can result in muscle spasms, confusion, paralysis, and even death. So this is really important to make sure that our bodies are maintaining the proper solute-solvent concentration. Malpighian tubules are the excretory organs of arachnids like spiders, ticks, and scorpions, and of insects like bees, butterflies, mosquitoes, and ants, and the list goes on and on. And so Malpighian tubules, they look something like these kind of filament-like projections that extend into the body cavity. So in this cartoon, you can see them as this purple, squiddy-looking material on the sides of the gut. And what they actually look like, you can see them here. So all of this yellow stringy material are the Malpighian tubules. So all of this are those tubules. And they extend from the sides of the gut into the body cavity, where they uptake potassium salts and waste from the hemolymph. So the waste and the potassium salts are actively being pumped into these tubules. And when that happens, due to the difference in solute concentration, water will also diffuse into those tubules via osmosis. And so once water, waste, and the salts are all mixed together, they will travel down the tubules into the gut. And once in the gut, the liquid waste will be combined with the solid waste. And then all the liquid and solid waste will travel down the gut. And as it's being traveled further down, water is being reabsorbed and the salts are being reabsorbed so that the animal doesn't lose those valuable resources. So they need the salt and the potassium salts and the water to survive. And so constantly more and more water and salts are being reabsorbed as you get closer and closer to the anus. By the time the waste meets the anus, the only thing that's left is the solid waste, so the feces, and a dry nitrogenous waste called uric acid. And everything else has been reabsorbed. And so this allows for the animal to conserve a lot of water because they reabsorb the vast majority of it. And instead of having water incorporated in their feces, they just release the kind of a dry feces instead. So this is the way that arachnids and insects undergo their excretory system. Vertebrates use organs called kidneys in order to undergo osmoregulation and also to filter waste from the blood like nitrogenous waste. And the excretory system in vertebrates is far more complicated than what we saw in the invertebrates when they used Malpighian tubules. And so looking at the kidneys, there are a couple of different major components. So we have our nephrons, which are responsible for doing the actual filtering. And they're in this lower image, this yellow um, a bunch of tubes. And then we also have blood vessels. So we have our arteries, which are shown in red, and then we have our veins, which are shown in blue. And so the arteries bring blood in to be filtered, and the veins carry the blood that's been filtered away back to the body to be reused, and now it's clean. And so the kidney also has a couple of regions to it. So we have our cortex, which is the outermost part of the kidney. And in this picture, it's shown as this more light pink area here. And in this image as well on the top, all this kind of light pink area is the cortex. And then we have the innermost part of the kidneys, which in this picture is this darker pink area. And it's also represented here in these kind of sacs here is the inner part of the kidney. And there's a difference in solute concentration in the interstitial fluid between the cortex and the inner part of the kidneys. So as you get closer and closer to the inner part of the kidneys, the solute concentration in the interstitial fluid increases, and this plays an important role in the reabsorption of water and salts and things like that. And we'll talk about all those stages in the next slide. And so the kidneys play an important role in filtration, reabsorption, and secretion. In the filtration stage, it's responsible for filtering out excess waste and salts and things like that from the blood. So blood comes into the kidneys from the renal artery, which is shown here as this red vessel going into the kidneys. And once the kidney reaches the nephron, and we'll talk about a lot more detail on what's happening in the next slide, but the blood reaches the nephron and it will become filtered out. So all of the water, salts, various ions, and waste will be removed from the blood and collected into a sac on the nephron. And once it's collected, it's no longer called blood, it's actually called filtrate. And then the blood cells and any large proteins will actually remain in the artery and go back to the body and leave the kidneys via the veins. And the main vein that leaves the kidneys are the renal veins, which you can see here in this blue in this image. And so this can be energetically expensive for the kidneys because they're going from a lower concentration of solutes in the blood vessel to a higher concentration of solutes in the nephron. So some of those solutes have to be actively pumped out. And so this requires ATP, so it requires a lot of energy to undergo this process. But there's also pores in those vessels that help some things move through freely. So even though it's energetically expensive, it is very, very important for the cells to undergo this process. Otherwise, you'd have waste just flowing around in your blood forever. And then as the filtrate is traveling through the nephron, a lot of needed water and salts and ions and nutrients are reabsorbed into the body so that we don't remove it as waste. And this process concentrates the filtrate. So as the filtrate keeps moving through the nephron and we're removing more and more water and certain ions and things, the waste that remains becomes more and more concentrated. And then the whatever's left that we don't need, so excess water, salts, waste, etc., is then super concentrated at the end and added to the urine and in the bladder, and then eventually it's released from the body when you go pee. And so now we're just going to go through each of these steps in more detail so you can understand how kidneys actually function. We begin with blood traveling into the kidneys via the renal artery. And then as the blood is traveling through the renal artery, it will become diverted into these offshoots of capillaries. And these capillaries lead to a dense, compact area of capillaries called the glomerulus. And so as the blood is traveling through these capillaries of the glomerulus, the water and salts and waste are being removed from the blood. And this is due to the high pressure and porous nature of the glomerulus. So the vessels in the glomerulus are highly porous, and then there's lots of pressure built up in those capillaries because the pressure of the blood flowing through the renal arteries is far more concentrated in the lower area, the smaller area of the capillaries. So this creates a lot of pressure in those capillaries that basically pushes out all of the water, waste, and salts from the capillaries. Now, the things that don't fit through those pores are large proteins and the red blood cells because the pores are too small. And so the large proteins and red blood cells will pass through the glomerulus and then travel to the veins and then are removed from the kidneys via the renal veins to go elsewhere as filtered clean blood now. And so the salt and water and waste that's removed is collected in this structure, the circular structure called the Bowman's capsule. And that is shown here. So it's all collected there. And then once it's collected, it's called filtrate. So it's no longer referred to as blood. This is now called filtrate. And the filtrate will move from the Bowman's capsule into the nephron. So it'll travel through the tube to the nephron. And as it's traveling through the nephron, it will start to pull out the sodium from the filtrate. And this requires ATP, so it's active transport of sodium. And so as the sodium is being removed from the filtrate, the chlorine ions will follow by diffusion due to differences in charge. And then also water will diffuse out via osmosis because now there's a difference in solute concentration. So this is our first step of reclaiming salts and water from the filtrate. And then the filtrate will continue to move down the nephron to something called the loop of Henle. And that's this area here. And as the filtrate is moving down the nephron is going from the cortex to the more inner parts of the kidneys. And as we talked about before, there's a difference in solute concentration as you go from the cortex to the inner parts of the kidneys. So let's say we're going to just use some dots. So let's say the solute concentration at this part of the cortex is relatively low. But then once we move to here, we have a higher solute concentration, and then higher solutes even more, and then even more solutes, right? So the higher, as we go and increase in solute concentration, the higher the solutes in the interstitial fluid, the more water will be removed from the filtrate into the interstitial fluid. So we're reabsorbing more and more and more water. And then eventually we get to the bottom of the loop of Henle, and it will start to travel up again. So the nephron will travel back up to the cortex, and while it's traveling back up, the nephrons are then going to actively transport sodium out of the filtrate. So more sodium is being pulled out, which will cause more chlorine to come back out of the filtrate, and this will cause the area of that area of the cortex to become more solute dense, which will cause more water to also come back out of the filtrate. So we're continually just reabsorbing salts and water throughout this entire process. And then eventually we get to the end where now we've absorbed a lot of water and a lot of salts, and the filtrate is far more concentrated than when we started at the beginning. So the concentrated filtrate will then join into the collecting duct. And there are several collecting ducts for each for each nephron has its own collecting duct, so there's several of them. And so the filtrate will then travel down the collecting duct, and the collecting duct travels towards the innermost part of the kidney again. So as the filtrate is traveling down that duct, more water is being absorbed. This is the last chance for the kidney to absorb any water. And so once we get down to the very end of the collecting duct, several collecting ducts will group together, and they will all funnel their urine into the same space. So now we no longer refer to the waste as filtrate, we now refer to it as urine. And so the urine from all these various collecting ducts come together in a structure called a ureter. And then the ureter leads to the bladder, where it drops off the concentrated urine. And then the bladder holds the urine until you go to use the restroom. And when you go to the restroom, the urine will pass from the bladder through your urethra and then out of your body. So that's the general stages of how this process works. I know it's a lot going on. If you need a more visual representation of what's happening, I've included a SciShow video at the end of this presentation that does a really good job of animating this process, and then just going step by step to understand how the kidneys operate. You've heard me mention nitrogenous waste a couple times in this presentation, and that's because nitrogenous waste is one major waste product that our excretory system is responsible for getting rid of. And if it doesn't do it properly, we can get very sick or die. And so nitrogenous compounds are a result of the breakdown of DNA and proteins. And this can occur from when cells naturally die. We break down the DNA and protein that made up those cells, and we can either recycle those parts or get rid of those parts as waste. In addition, just regular healthy cells, they're constantly making new proteins and breaking down old proteins. And when they break down those old proteins, it makes a nitrogenous waste as well. The primarily type of nitrogenous waste that is a result of the breakdown of DNA and proteins is ammonia. And ammonia is bad news for an animal because it's toxic to our cells and our tissues. So we have to figure out ways to get rid of it as quickly as possible. For aquatic invertebrates like lobsters and crabs, they're able to just release their ammonia directly out of their body into the water. So this is good for them because they don't have to expend any energy to convert the ammonia into anything else. And they also don't have to worry about like diluting it or anything like that. They can get rid of it very quickly, just expel it from their bodies and walk away. But if we as terrestrial vertebrates, or if any vertebrate, even aquatic vertebrates, wanted to get rid of their ammonia in a similar way, we would have to carry around with us extra gallons, just gallons of water to dilute the ammonia out to non-toxic levels. And that's not practical, especially for terrestrial animals. We don't want to be lugging around extra water just to dilute our waste. So instead, what we do is we convert ammonia to less toxic compounds. And in vertebrates, we convert ammonia to a compound called urea. And so you can see this is ammonia here. This is the structure, and this is the structure of urea. And we make urea by combining CO2 with ammonia, and the new compound of urea is far less toxic to our bodies. So we can store it in our bodies for longer periods of time, and we can also store it at higher concentrations than you can store ammonia because it's not nearly as toxic. So if we can store it at higher concentrations, that means we can also conserve water. We don't have to lug around gallons and gallons of extra water to dilute out urea because it's relatively non-toxic to us. But even though there's benefits to this process, it is energetically expensive because in order to build compounds, you always have to put energy into the system. So our bodies have to use ATP to convert ammonia into urea, but it is very important. So it's worth expending that energy to do. In terrestrial invertebrates, like insects, they make a different nitrogenous compound called uric acid. And uric acid looks like this. And so as you can see, this compound is much bigger than urea. So to convert ammonia into uric acid, the invertebrates actually have to expend more energy to make this larger compound. But on the opposite end, they do get a major benefit because they can conserve even more water because uric acid is even less toxic than urea is. So they need even less amounts of water in order to dilute out their nitrogenous waste compounds. So there are some pros and cons to both urea and uric acid, but no matter what, we cannot keep ammonia in our bodies. So we have to convert it in some way. Alrighty, so that's the end of our lecture on some basics of animal physiology and then the homeostasis and the excretory system. So go ahead and read chapter 21, sections 1 through 10, and then 12 through 13, and then do the learning curve assignment for chapter 21. And then I've also included here 2 videos that I think are really helpful. The first is a crash course video on the excretory system. And so this is the video I was telling you about that will show you a more visual representation of how kidneys work to help make it a little bit more sense. And then another video here on the 4 tissue types. Both these videos do have some excess details that go in a little bit more depth than what we're covering in the class, but I think it does, they both do help to understand the concepts really well and to visualize those things. So I recommend these videos. And of course, any other videos and tools and resources that you find online that help to submit these concepts is always a good resource. And so with that said, that's the end of this lecture. See you next time.